Rajiv Saxena, Associate Professor, Smriti College of Pharmaceutical Education, Indore. In today's video lecture, we'll discuss about animal cell culture technique. In the last video, we have gone through the introduction of animal cell culture technique, and we have also introduced various terminologies which are related with the animal cell culture technique. We have also discussed about various application of this method and also cover various nutritional requirement that is necessary for the growth of the animal cell culture. So that day we have discussed that for the cultivation or for the growth of the animal cell, different medias are actually available. These medias are actually classified into two broad categories. Number one is a natural media and number two is a synthetic media. Natural media generally consists of the plasma, serum, tissue extract, whereas the synthetic media generally composed of various organic and inorganic nutritional supplement along with the serum proteins, carbohydrate, vitamins, etc. So, in today's video lecture, we'll discuss about how we can proceed the animal cell culture technique in our laboratory. So, the first step, suppose if we have to perform the animal cell culture technique in our laboratory, the first step is actually the isolation of the tissue. So, we know that we are cultivating the cell. So, the cell cultivation starts from the collection or the isolation of the tissue. So, the tissues are generally collected from the experimental animals, maybe your rat, mice, guinea pig or maybe mouse embryo. Otherwise, we can also go for the collection of these tissue from the human body also, like we can go for the collection of the smooth muscle cells, we can go for the collection of the macrophages, lymphocytes, etc. So these tissues are actually collected from the different sources, maybe from the animal sources or maybe from the human sources. But once this tissue will be collected, we have to properly wash the tissue, and suppose if we have to keep the tissue for some time, it is generally kept in the BSS solution, which is basal salt solution, in which some antibiotics are added in order to prevent the growth of the microorganism. So the first step is the isolation or the collection of the tissue, and then we have to go for the proper washing of the tissue. We have to keep the tissue in a BSS solution in which we have already added certain antibiotics. Now, coming to the next step is the disaggregation of the tissue. We know that the tissue is actually a larger mass of cell. So if we have to go for performing the animal cell culture technique, what we have to do? We have to convert the tissue into cell. So for that we have to go for the disaggregation procedure, in which the tissue is disaggregated, and the disaggregated tissues, from the disaggregated tissues we can go for easy collection of the cell. So for the disaggregation process, two methods are actually available in our laboratory. Number one is a mechanical method and number two is a enzymatic method. Suppose if we are performing the physical method, that is the mechanical method, so under the mechanical method, again different processes are available, like we can pass the tissue with certain sieve, we can pass the tissue with the help of the syringe, or we can go for the repeated pipetting method. So by this physical method or by this mechanical method, what we are doing, we are trying to reduce the mass of the cell, we are trying to convert the tissue by mechanical forces into the cell. But again, this method is having certain disadvantages, because here mechanically we are trying to disaggregate the tissue, so somehow this process can also damages our cell. So, some alternate methods are also available, and the best practice method in our laboratory is actually the enzymatic disaggregation process. We know that the tissues are actually attached, the cells of the tissues, they are attached with each other with certain fibrous proteins, maybe collagen. So here what we are doing, we are trying to disaggregate or dissolve the cementing material which is available between two cells with the help of different enzymes. So under the enzymatic method, of disaggregation, two enzymes are commonly used. Number one is a trypsin and number two is a collagen. So, trypsin, if we are using the trypsin, the process is actually called as trypsinization. The trypsinization process of enzymatic disaggregation is again divided into two categories. Number one is the hot trypsinization and number two is a cold trypsinization method. In case of hot trypsinization, what we are doing, we are first collecting the tissue, okay? Like in the last step, we have already collected the tissue, which is already kept in the BSS solution along with the antibiotics, okay? So what we are doing in case of the hot trypsinization, it doesn't here mean that we have to heat the trypsin. Here, the hot term is actually associated with the temperature, which is around 36.5 degree centigrade. So we have to keep the tissue in which we have added the trypsin and we need to maintain the temperature of around 36.5 degree centigrade. So under this condition, the tissue will be kept for around 4 to 5 hours. After that period of time, the trypsin will be removed and we have to go for the addition of the serum containing media and then we have to go for the repeated pipetting method. Okay. So what will happen? Because of the action of the trypsin, the cementing material which is available between the cell will get dissolved. So what will happen? The tissue will become very fragile in nature. So suppose if we go for the repeated pipetting of that tissue, then the cells which are available in the tissue, they are very loosely arranged because of the action of the trypsin, and then what will happen? This will be easily disintegrated with the help of the repeated pipetting method. So here we are using the trypsin and the temperature of this particular process is around 36.5 degree centigrade. So that's why we are calling it as a hot trypsinization method. The method is continued. Suppose if we are performing, either we are performing the hot trypsinization method or cold trypsinization method or maybe the collagenase treatment method, then the process is actually continued until we'll get around 10 to the power 4 number of cells per ml of the solution. So, the second method which is available with the help of the trypsin is by the cold trypsinization method. So again what we have to do, we have to take the tissue that we have collected in the last step, then what we have to go, then we have to go for the addition of the trypsin. But once we go for the addition of the trypsin, what we have to do, we have to keep this particular vial over the ice bath. So that's why here the trypsin is available in the colder condition. That's why this method is actually called as cold trypsinization method. So over the ice bath or over the ice, the vial will be kept for around 4 to 6 hours. After 4 to 6 hours, we have to keep the vial again at around temperature of 36.5 degrees centigrade for 30 minutes. Then we have to go for the removal of the trypsin. Then again the serum containing media is added and again the repeated pipetting procedure is followed like as in case of the hot trypsinization method. So again what will happen? Because of the application of the trypsin, again the cementing material available between the cell will get dissolved under the cold condition and further the fragile mass can be disintegrated by the procedure of repeated pipetting method. Now otherwise, if we are not using the trypsin in our laboratory, we can also go for the use of another enzyme which is called as collagenase. It is considered that the treatment of the collagenase is much easier as compared to that of the trypsin. So again in this method also the same procedure is allowed. Okay, what we have to do, we have to take the tissue, then in that tissue here in this case we have to go for the addition of the enzyme which is collagenase. And after the treatment of the collagenase, again what we can do, we can keep this particular tissue mass on some stirrer, and so that the continuously the collagenase will be in contact with the tissue. It will try to dissolve the cementing material, and time to time we can withdraw the cell from this stirred condition, and again we can go for the analysis of the number of the cell, and once the number of the cell will reaches to 10 to the power 4 cell per ml of the solution, till that we have to go for taking the or harvesting of the cell from the tissue. So almost the method of the trypsinization method and the collagenase method, they have the similarity that because of the addition of this enzyme, what we'll get? We'll get the disaggregated mass of the tissue. So once the disaggregation procedure will be stopped or will disintegrate the tissues, then what we have to do? We have to go for seeding the culture into the culture vessel. As you can see in this diagram, initially we were having some tissue, and because of the trypsinization or collagenase treatment, we have converted that tissue mass into disaggregated cell. So this disaggregated mass of the cell is further allowed to introduce into the culture vessel. So specialized culture vessels for the animal cell culture is available in the market, and generally these vessels are either made up of glass, it can be made up of plastic, and some metallic animal cell culture vessels are also available. But most commonly the plastic culture vessels, disposable plastic culture vessels, they are widely used in the laboratory. So here what we are doing, we are actually seeding this particular disaggregated mass of the tissue which is converted into the cell inside the culture vessel. So in the last video, again we have introduced one term which is called as anchorage dependency. So it is observed that suppose if we are talking about various human cells like the cells of the kidney, the cells of the liver, they are having the tendency in order to grow by anchoring with the base of the culture vessel. So as you can see in this diagram, the cells are actually anchored at the base of the culture vessel. So it is considered that the cells, these particular cells, they have some positive charges, and the vessel that we are using for the cell culture methodology is having certain negative charges. So because of that positive and negative interaction, the anchorage dependency will take place. But again we have to see in this condition, like there are certain cells which are non-anchorage dependent, or they are also called as anchorage independent. So in case of the anchorage independent cell, this animal cell culture is available, or the culture will take place in the form of some suspension. Okay. So here as you can see in this diagram, whatever the disaggregated mass of cell we have collected, we have introduced that particular mass into the culture vessel, and we are providing the condition so that the anchorage dependency property can easily be followed in this particular cell. Sometimes what we do, sometimes we can go for even the treatment of this culture vessel. Suppose if the culture vessel it is made up of plastic, or maybe it is made up of certain glass material, so in order to enhance the anchorage dependency of the cell so that they can be easily anchored at the base of the vessel, we can also go for the addition of certain substances like some gelatin, some fiber, some collagen material. They are actually spreaded at the base of the culture vessel so that the anchorage dependency property can be increased because of the treatment of the substrate. So then what we have to do after the introduction of the cell, then what we have to do, we have to go for the addition of the culture media. Either we can go for the addition of the natural media, or we can go for the addition of some synthetic media, like for example RPMI media. Now once this vessel will be ready, it will have the cell, it will have the culture media available, then definitely the cell will start multiplying. So this culture vessel is again kept in certain incubator like CO2 incubator, so that the growth of the cell will takes place. But we also know that these particular cell, when we try to cultivate this cell under in vitro conditions, then it is also having the property of contact inhibition. It means once the monolayer will be formed in the culture vessel, then if suppose if we'll keep this vessel for the further period of time, then what will happen? Then maybe because of the exhaustion of the nutrient components which is available in the nutritional media, or maybe because of the generation of various toxic element, the cell growth will be hampered. So meanwhile, what we have to do, we have to go for the subculturing of these particular cells. So the subculturing phenomena, like we have discussed in case of the microbiology, we have to go for the subculture of the microbial cell. So here also we have to go for the repeated subculturing of the cell, so that every time we can maintain a very long-term continuous cell line. So a very important technical term that is associated with the subculturing here in case of the animal cell culture technology, it is actually called as passaging. So we have to go for passage of the cell that are already cultivated into the culture vessel. Once we will find out under the microscopic condition that the entire floor or the entire substrate or the entire base of the culture vessel is actually occupied by the cell. So we know that now it is exactly the correct time when we have to go for the subculturing of these particular cell. So from that we can go for the obtainance of the primary cell culture. Once we'll get the primary cell culture, from the primary cell culture, every time when we go for the passaging of the cell, then we'll get the secondary cell lines. From the secondary cell lines we'll get the tertiary cell lines and so on. So in this, by using this particular cell lines, different studies can be performed. We can easily go for the production of various biologicals, and so many applications of this animal cell culture technology is actually available that we have already discussed in the last video. So summarizing here, these are the basic steps that we need to follow if we have to perform the animal cell culture technology in our laboratory. First we have to go for the isolation of the tissue, washing of the tissues, then keeping the tissue in BSS solution along with the antibiotics. Then we have to go for the disaggregation of the tissue either by using the physical method or by using the enzymatic method. Once the disaggregation process will be over, then what we have to go? We have to go for the seeding of this particular cell into the desirable culture vessel. Then we have to go for the introduction of the culture media. Then we have to keep it for incubation, and then the repeated passaging is actually performed so that the continuous cell lines can easily be obtained in our laboratory for long-term experimentation. Thank you so much.